Professor Pawan Dhar from School of Biotechnology, JNU, and we have with us our DIA, actually Head of Department, Professor Ramakrishna Muzurika. We have with us Professor Anjana Munshi and other faculty members in the department, Dr. Ravindresh, Dr. Vinay, Dr. Shashank. And herein I request Professor Ramakrishna sir to please say some opening words for us.
So good afternoon, everyone. Welcome to all the faculty, colleagues, and all the students who are participating, and our Honorable Vice Chancellor, who always encourages us to conduct webinars which are of academic interest and research related. So today we have Professor Pawan Dhar, who is professor in the School of Biotechnology at Jawaharlal Nehru University. So the topic he's going to speak is something very interesting and quite different. Okay, synthetic biology. So he has a rich experience that I can see here in terms of his publications, research funding, and all other academics related experience. So this is a great opportunity for many of our students to learn about research, what he's doing, and especially synthetic biology, and in general about research, how to go about research and do a productive research. And so hopefully all our students and also my colleagues, we'll all learn a lot from him. Dr. Manju.
Thank you so much, sir. So herein I would like to give a brief introduction based on a long CV of Professor Dhar. So he presently heads synthetic biology group at the School of Biotechnology, JNU. He is former dean of the same school at JNU. As we mentioned, he's an expert on synthetic and system biology, wherein we have synthetic molecules. In fact, the concept extends to the minimal synthetic cell, even tissues and organs. So that way, the area holds a great transitional potential, wherein there can be reprogramming of the biological processes going on. So in that context, a lot of basic and implied importance of the area is very apparent. I would like to mention that Professor Dhar has immensely contributed towards development of synthetic biology platforms in multiple places of academics and industrial relevance in different countries, India, Japan, Singapore. And he is into many very different activities, which is obvious with his landmark publications, multiple patents. And yes, it seems that he is actually towards going in the direction of, and has already contributed immensely towards shaping up the entire field of synthetic biology per se, with his futurist insight. He is already a co-founder and chief mentor in context of a biotech company, and the name itself suggests this the foresightedness, Foresight Biotech Private Limited. So with this short introduction, I'm not going into the details of the kind of papers, publications, and the projects. Yes, so the kind of projects are quite interesting. As I was going through the CV, in context of developing the very field of synthetic biology, like capacity building for marine synthetic biology in India, policy and research planning for synthetic biology. So that says a lot. And I think I should be offering up the platform to Professor Dhar without wasting any more time. Please.
Thank you. Thank you so much, Manjuji and Ramakrishna sir and Anjana ji. It's a pleasure meeting you after such a long time. From Jammu University, more than 3 decades, and really looking forward to coming over there sometimes in future.
Sure.
So, thank you very much for your kind invitation. And today, well, I would like to give a little bit of overview of the field, the way I've learned it, but also my personal journey with this. When I was doing my PhD in Banaras Hindu University back in '92, this was in the area of human genetics. By the time I finished the PhD, whatever work I had done in chromosomal aspects of the tumors, all of that got obsolete because the science moved so fast. We just could not continue that. So we have to jump onto, fluorescence in situ hybridization, chromosome painting, go deep into the molecular aspects. And the boundaries, the cutting edge is changing all the time because people work very hard to innovate. So one has to keep learning. And then after I joined my faculty position, the first one was at Manipal, where I said the Human Genetics Division. I used to go part-time for software engineering classes with Aptech. For 2 years, I learned software development after doing PhD in genetics. Then bioinformatics was launched, so it came very handy, all this knowledge. And then the fields kept on changing as and when the questions changed, the employment places changed. And so in Japan, it was more of gene knockout, DNA repair, and then systems biology, building a virtual cell. And then synthetic biology happened. I was in US at that time. This is 2004, and there was a conference in MIT, the first synthetic biology conference, where the field itself was announced. So I was very fortunate to be there, a 3-day event, and it was a wonderful realization. So whatever systems biology work we were doing, I just stopped it. It didn't make sense compared to what was coming up. And then we also launched a new journal with Professor Ron Weiss, who was at Princeton University at that time, 2007, and then joined MIT. And we ran this journal for some time, 7 years. So today, my purpose is to give you an overview of what is synthetic biology, why synthetic biology. Because if you ask even now the practitioners of synthetic biology, not only in India, but also overseas, you will get different definitions depending on whom you're talking to. A biologist, a chemical engineer, a mathematician, or an engineer, from the engineering background, a mechanical engineer, let's say, or electronics engineer. They have different ways of looking at the same theme. So my humble attempt first would be to impress upon you the need to have synthetic biology as a new discipline. And then I'll quickly move on to our work and then leave you with some unanswered questions. And I hope that we can find some common grounds in the process. So I'll switch off my video because my bandwidth is giving me trouble sometimes. So please excuse me, but then I'll switch on the slides now. Just give me 1 second. Can you see this screen, synthetic biology?
Yes, sir.
Is it visible?
Yes, Pawan,
Okay.
it is.
Yes, sir.
Okay, sometimes what happens is that, the slides go, without video or without audio. So if that happens, Manjuji, please do call me on my mobile phone because this often happens.
Sure. Yeah.
Okay. Thank you so much. All right, so this image essentially captures the essence that we take biological information, use the engineering principles, do a bit of modeling, and go back to the experimental phase. So why are we talking about this, and what is the beginning of all of this? You see, when you look at the way science has been practiced, it is mostly reductionism as a way of doing science. Reductionism means that, for example, Mendel thought of a pea plant in terms of the causative factors and went from genotype to phenotype and phenotype to genotype. So it was like a zoom in, zoom out. So from the phenotype, he had to imagine some fundamental elements that were creating the phenotype. So he went all the way up to the factors, imagined them without seeing them. So this process of going from the gross to subtle is called reductionism. So if you have to explain how an organism behaves, you've got to open it up. So when you open up for the first time, let's say an eukaryote, you call it gross anatomy. When you go a little deeper into the gross anatomy, and this is what you see. When you go a little deeper, you come across the microscopic images of the tissues, and you call it histology. When you go a little deeper in histology and you see cells like this, and then in a normal staining method, they look very innocent, they don't have much information. But as in the microscopic technology and the staining procedures, when they become more sophisticated, you have differential staining and high resolution of microscope, you see more structures within the cell, and this now becomes a new field called cell biology. When you zoom into cell biology and you go to the level where you now identify individual components in the organelles, and one of the star components has been chromosome for obvious reasons, and it's made of DNA and histone proteins, as you all know. DNA coils itself and uncoils 7,000 to 10,000 times to make a chromosomes and so on. Now, DNA was the megastar during early 30, 40, 50, 60s, and 70s. And so people used to consider DNA more as a director, which is controlling everything. But then the RNA and protein community caught up in the subsequent years. This is the beauty of science. And then nowadays, we consider DNA just as a database, which has got some information out there. All right. So when you go beyond this in the reductionist process, you come across molecules. Now you have left the domain of sort of biology. You're getting into the domain of chemistry. Now you think of chemical molecules, organic molecules, chemical bonding, torsion, angles, and all binding forces. When you go beyond this and you say, what are these molecules made of? Remember, we started with the pea plant, or in this case, an eukaryote, an animal. You're going all the way down to explain a certain behavior. Beyond molecules or beyond this chemistry layer, you come across the layer of physics, the atoms. Now, when you want to go zoom in further, what you see is that it is 99.9% space. So this is very interesting here because you don't have a method to stain the space or put GFP in the space, because all the knowledge that you gathered so far was because of the light colliding with something. When the collision doesn't happen, you don't get the information. That data cannot be retrieved. The light has to collide with something. So therefore, I think I'm getting some echo. If you could switch off the mics. Okay. So, so now, when you are, trying to go to this level, you lose the resolution. So it's better to go one step above and stay at the level when you can actually see the molecules in this reductionist process. So this defines sort of the limit of reductionism. And here it's more of fundamental physics, and then atoms talk to each other and chemistry is born, and chemistry happens in the biological setting, and we call it biochemistry. So the whole process that we just went through, the reductionist process, it gives rise to molecules. So for example, if you just take a cell, not an organism, in terms of eukaryotes, but a single cell organism, let's say a microbe, and you do a reductionist processing of this structure, what you get is various molecules in terms of genes, RNA, proteins, and then are lipids, then there are carbohydrates and water molecule, and then so on and so forth. There is so much to say about it. Now, when you reach a stage when you have a lot of data, and now the challenge is not to get the data, but to make sense of the data. So what you have done is using your logic, using the reasoning, you develop technology. And this technology allowed you to go in a reductionist way. You're chopping at every level. You are chopping and getting information, chopping that and getting another information, finally reaching up to the level of molecules. Well, the reductionist process has been highly successful. That means the chopping itself has been highly successful, and we got all the molecules. But the original object is gone, all right? So the original organism is gone. You all, the entire information that you have right now is a bunch of molecules. Now we have to make sense out of it. When you have thousands and thousands of them, unlike Mendel, he had to use the notebook, but now we are talking of thousands and thousands of them. Very large search space. What you do now, you use computers and you create databases. So this is another way of doing science. First was reductionism. The other is database. In database, you build models, and database itself is a model of the system. but you build pairwise interaction and pathways and so on. These are two different ways of doing science. One is reductionism, and another is integration. If you take integration a little bit further, you can build virtual cells, virtual organisms, and so on. Third approach that engineers started back in 2004, the meeting that I was referring to a while ago, they asked, can we construct a chromosome? Can we chemically synthesize a mitochondria? Can we chemically synthesize ribosome? Can we chemically synthesize genome? Well, we know that primers can be synthesized. Now, can we extend this, the whole story of synthesis, and can we synthesize or boot up the whole cell with the chemically synthesized, versions of molecules? So this way, it encompasses not only the philosophy of construction, but also standards and rules of composition. So the first approach we popularly call as molecular biology and biochemistry, or you could call histology and cell biology and so on. The second approach, if it is single molecules, we use the term bioinformatics. If it is a pathway collection of molecules or the group behavior of molecules, we use systems biology. And the third is what is called a synthetic biology. So I hope you got the terminologies right. What is the need to start a new approach? Some people might say that you are just renaming molecular biology as synthetic biology. So what's the big deal? Now, big deal is that when you're trying to construct something, now you might also say that, well, we have understood construction in terms of genetic engineering. Well, genetic engineering is a nice word, but we never engineered genetics. It was all a probability. So you take, let's say, recombinant vectors, thousands of them, and you put them over millions of cells, typically, let's say, 10 to 20 million in a multi-well plate, and then you make sure there's some electroporation effect, and there's a possibility of vectors finding their way into the host cells, whatever they are. Now, this entire process is a probability, because after all this nice process, you might get maybe 6, 8, or 10 colonies out of thousands of possibilities. Now, this is a probability. This is not engineering. Engineering would be that you're absolutely certain that you're doing a reaction and it's leading to some effect. So you're adding a lot of accuracy to the whole process. So this is the idea I gave many years ago, because we have a fantastic reductionist technologies and a lot of data at the level of DNA, RNA, protein. Is it time to think of a periodic table in biology? And if yes, what would qualify as an element of this periodic table? So there was some thought experiment, and we felt that fold would be an appropriate member of this bioperiodic table. The trouble is, this bioperiodic table is still a theoretical construct because it doesn't have any physical basis, because there are a lot of information gaps upstream and downstream of the fold. So it's still going to be a theoretical construct for a long time. So the question is, can we think like an engineer? Why are we thinking like engineers? Is because there is a lot of similarities between biological and engineering systems, as you can see clearly on the screen. However, there are also dissimilarities, especially in terms of the biology. We are dealing with mobile components, and while as if you use computers, you have transistors and wires and switches and so many components. They are fixed to the motherboard in terms of computer, let's say. So there's an anchored component and there is a mobile component. There's a big difference because your information is not programmed to move in a certain trajectory or a pathway. It is going to be there in a certain space. It's a constrained space, but it is not hardwired the way it is wired in engineering systems. Likewise, biological systems are predominantly analog. They're not digital. Digital would be that you switch on let's say, electrical bulb and you switch it off. Analog would be that you move the fan regulator from 0, 1, 2, 3, 4, 5 at different speeds. You can move the fan. This is analog system, and that's the way. That is how biological systems are. And Mendel's laws, if you call them laws or if you call them ratio, that's fine. They were not designed to engineer things from scratch. They were only They were only there to tell us something about the system, but they were not there to design things from scratch. So they're more of, you're looking at from phenotype to genotype and genotype to phenotype, and the layers in between are totally abstracted, but that's where we have to be. So what do? Biological systems are noisy and unpredictable. You know about the gene expression noise and protein expression noise. Noise means in terms of the fluctuation. So it's not as I said, the digital switch is not there. It's an analog system. So this analog system has a variation of the concentrations. At every given moment, the concentration will be fluctuating. So it's very difficult, or shall I say impossible, to say that this point in space represents gene expression. No, it's an entire range of the gene behavior that one has to consider. If you look at the parallels between an electronic circuit, we can transfer the information through predefined nodes. We call them as logic gates. But then in biological circuit, it has to come one step down, that you have to make mRNA, from there you have to make protein. And it's a little more complex because mRNA gets degraded. And if enough mRNA stays there till the level of protein, and then enough protein is available and it does not get degraded, and then it is nearby the transcription factor binding site, then only the next switch event will be switched on. That's not the case with electronic circuits. It's pretty straightforward. So that's why it's difficult to create biological circuit equivalent of electronic circuit. So to design well-behaved system, one needs standards and rules of composition. And standards are something like this. When you have the rails are fixed at a certain distance from each other, it does not mean that it has to be 5 feet 6 inch all the time. You can have narrow gauge, you can have meter gauge, you can have different versions. But for that particular model or design, the standards are fixed. So standards do not mean accuracy. It just means that this is the way it is. It has been derived after some logic. Maybe early on, when these standards were developed in the UK, maybe when they were clearing the forest, the amount of space that was available was comfortable for this particular width. So they say that, okay, let's make the rails and the vehicles that are running on these rails, the train locomotives, let's design them based on the space that's available within the forest. So they certainly made a certain design. And in India, we just followed that. We did not have forests, but we didn't have to clear those forests. Even if it was a barren land, our standards were the same because we are importing those standards. So one more thing here, the standards have to be transferable, right? That's the nature of standards. So then along with the standards, you also have rules of composition. Rules of composition means that in what order do you compose these particles or these parts in order to build a certain structure. So your rules of composition and standards have to be fixed. The grand challenge is really to engineer biomolecular and multicellular circuits using construction standards. But for standards, you need constants. You cannot have variables, right? So where do you get these constants? You have a sample of E. coli constants, as you can see. Certain things are absolutely constant, something are relatively constant, and most of the time it's variable. So when you have a variable space to negotiate, it's very difficult to build standards. Nevertheless, because there is a payoff, it's always a good pursuit. So IEEE standards do not exist in biology because organisms were not created to solve human problems. Therefore, it's good to use engineering as a philosophy. It is good, but in practice, one has to be different because they don't exist. Probably engineers never faced a problem as complex as biology. What does that mean? It is not to downplay their contribution. It is just that in biology, you have emergence. Engineering systems, you don't have emergence. Emergence means that when molecules are single, they're behaving in a certain way. When they're together, they're behaving differently. For example, not only molecules, it can be the entire organism. When organisms are single, they behave in a certain way. When they are together, the behavior changes. For example, the school of fish that you see in oceans, the way they move in certain patterns, you cannot make out that pattern by looking at fish. Or the flock of birds that you see, you cannot predict this flock, the design, the way they change the pattern by looking at the birds. So that's a property of the living systems. Also, you have probability and gradients. So what's the solution? Well, it's a practical solution rather than an absolute solution. In the traditional setting, we have been good in decomposing the system at one part at a time. In the new setting, synthetic biology approach, we compose it, but we have to make sure it's a controlled construction. Otherwise, you start making E. coli and making Ebola. So one has to be very careful when you're constructing. It's an additional responsibility and a power. So terms like truth table have made their appearance in biology. What is a truth table? It's basically a logical table that tells you the state of the system in a certain input condition. So here, if the input is about inducer or repressor concentration and the output is protein concentration, you figure out what will be the state of the cell in that particular time. Works beautifully when it comes to thinking of engineering as a philosophy, but in practice it does not work because your truth table is not transferable. Even in the same organism, your truth table changes all the time because gene expression is fluctuating. So you cannot say that this is my protein concentration, it is fixed. No, it cannot be fixed because it will depend on the metabolic state of the cell culture conditions, and also the cell strain. So these are not transferable and these are not dependable values within the same strain. So that means this approach of engineering, though it looks very attractive, is not going to work in biology. Overall, synthetic biology is a rational design and controlled construction of biological components leading to applications. Early on, I remember when the term was announced on this was a 3-day event at MIT, and the third day, last session, Drivendi, who was the person who was supporting this, starting the synthetic biology as a new discipline. So he asked people, would you like to suggest a new name? Well, people started thinking because till then people are not very certain as to what is coming up. And so people did not accept the term straight away. There was a lot of discussion for several years. They did not like this term because they thought synthetic means something like a chemical, is a synthetic chemistry. So they did not straight away accept it. So there were a lot of discussions. And over time, the term biological engineering came up as an alternative. Well, people suggested constructive biology as a replacement for synthetic biology. But then people said, well, if biology is constructive, then is the rest of the biology destructive? So you cannot use this term. They said, okay, let's call it biological technology. If it is biological technology, then what is biotechnology? So then it came to biological engineering because it does not talk about, it's agnostic about the component size, which means it can start from the parts, devices, circuits, cell, tissue, and multiple organisms. It can go all the way from molecules to organisms. So that way people are comfortable. So if you come across publications that mention biological engineering and that mention synthetic biology, they both mean the same thing. It's just individual preference that matters. So there are companies using synthetic biology approaches to make components for the industry. OPEX Biotech, likewise Metabolics company. So where are we placed? Back in 2010, when I came from Japan, we started this event. So my biggest challenge was to find out who is interested in synthetic biology. Then we got the community together from IIT, from ICERS, from central universities. So we got people together and we created a new group called Sinjeevani. This was the name given by one of the PhD students from ISC Bangalore. Sin as in synthetic biology and Jeevani as in biology. So after that, there was another event that we created here. And then there was another event, and then there have been many meetings and so on. Also, the teaching initiatives began from 2010, offering 2 credit, 3 credit, 5 credit course in synthetic biology. And we recently finished a 3-year program from DVT when we trained people in synthetic biology for 6 months, and providing the scholarship, providing the accommodation and travel and everything was part of the package. So we also completed that, trained 45 students in 3 years. And it has been going on in a certain way for the outreach. But the question that we asked back then when I was in Japan, where the whole journey of synthetic biology began, So you see, we asked that, well, first of all, the observation before the question, that by converting the genes into junk and garbage, we have learned biology. That's how we learned. Junk is essentially something that it's not functional, but you don't throw it away. You know, for example, there's a broken chair or a broken fan or mobile phone or something else. We said, okay, maybe we'll use it sometime in future, so let's not throw it away. But it's not functional, so it's junk. But every day in kitchen, we throw the garbage, you know, in the form of vegetable trash. So that is a garbage. So that's how we learn biology. The question that we asked was, given that there are three types of sequences, RNA coding, protein coding, and non-expressing or not coding. It's not non-coding, it's not coding at all, which is what we call as the dark matter of genome. Can we make something from here? So this was also coming from the observation that nature placed these signals at a certain spot, and we call them as start and stop signals. The question is, why did nature place the signals at that spot in the genome? How did she know that my transcription will begin here and end at here? Did she try all the combinations and say, okay, now I'm happy with this combination, and that's what is available for the organism? Or was it a random event creating a certain product and then the organism found a use case for the product? What happened? We don't know that. But if we are just considering the possibility that nature did not make all the combinations, did not do all the experiments, then we are talking of information science when you consider this sequence as information, and then you say that from the same sequence, I can create more variants. In other words, if you physically place the start and stop signals at different spots, then let's see what happens. If you are to prove this, that you can make your own genes in the lab, then it's very important to, first of all, be harsh with your parameters. So we have to be very, very selective about the sequences, because once you make them, people will say, hey, this is already existing, so what's the big deal? So we chose intergenic sequences, 2,500 of them, downloaded this from the database, and then we converted them through genetic code table into the protein sequence, equivalents of E. coli genetic table. So we converted that into the polypeptide sequence equivalents, and then we matched all these proteins against every known protein in the NCBI database, the non-redundant protein database. Wherever we found similarity, we removed that sequence. We don't want that. The second filter was that we also removed sequences that were conserved. They were not found in the protein database, but they were conserved. We removed that also, because somebody would say the fact that they are conserved itself means that there's a use case. So how can it be proven that it's a brand new gene in the lab? So we took the sequences, as you see the red colored line, different directions, different sizes, overlapping, non-overlapping. We did not know what worked, so we just took it randomly. At this point, there were a number of odds against the idea, because it was the first such attempt, and there were other issues. So when I talked to my students, my postdocs and PhD students in Japan, I was working in an organization called RIKEN. This is RIKEN. And this place is famous for, they have invented a new element for the periodic table many years ago. And also they have the facility for the largest protein structure determination. So there were something like 42 NMR machines in the campus. So when I spoke with my Japanese students, well, they're a bit careful about their decisions. So they said, well, this is the first time we're hearing the possibility of making a functional gene from an intergenic sequence. So they refused to work on this project, because they were happy with their own biosensor and other synthetic biology projects that was going on. So they said, well, this is a new idea. So of course we respect their decision, because at the end of the day, it is they who have to work in the lab. So we said, no problem, you can continue with your work. So I had a technician from Singapore by name Chosu, and she's the one who carried all the experiments. And when she was successful, a lot of people jumped in. So now we have, of course, the story has matured quite a lot. But initially it was quite tough for us to convince people that this makes sense. So the question is now, how do you make a gene out of a non-expressing DNA? This is a very simple question. It's a very, very simple problem, undergrad level problem, because you know what is a gene. So if you install signals up and downstream, you've eventually made a functional gene out of a non-expressing piece of DNA. Of course, you need to take care of these triplets and all that. That's a constant, but that's still a very tiny aspect. But then if you are playing with the genome sequence, you know, there is a lot of addition of these elements. Anything can go wrong, both in the spacing, the mutation, and the placement. A lot of things can go wrong, and it will take a lot of time to reach this level. So we said, though it's attractive, it's not doable, it's not practical. And we are pitched against people who are regularly publishing in Nature and Science, the entire issues of Nature and Science. So we were quite worried that we had to do something which is going to be, practical. So we made these genes outside the genome. So we were looking for a vector which we could design. Fortunately, at that time, Invitrogen had released a PBAT topo. And it was like a God-given, gift for us, because this vector had been manufactured with an ability to provide promoter sequence, ribosome binding site, start codon, stop codon, and the icing on the cake was the epitope. Why was this so exciting? Is because now it's a his tag. So you have anti-his tag, anti-his antibodies. So which essentially means that when you express your protein at the Western blot level, if you're able to see the band, then you say, hey, this is my protein. It has to be my protein because you've been so stringent. If you do not see the band, then it's not your protein. So now it becomes much more easy problem that you have to do bioinformatics, you have to select the sequences, you have to amplify them, and then you insert them in the vector, express them, and there you go. So the big moment came when I remember one day when the results were about to come, and we were like gathering around the table in the lab, and we were like watching nervously. And finally, around lunchtime, we got these results. And in presence of the inducer, there is a protein expression. The minus indicates that in absence of inducer, there is no expression. We call them as eka proteins at that time. Eka derived from ekam, which means first, in Sanskrit. So we said that, these are the first such proteins that we have made, and we were very excited. Now the nature of questions changed. What is their structure? As I mentioned a while ago, we had these NMR machines, but then none of them is available for our work. So because they are running with the Protein 3000 project, 3,000 proteins, they have to do the structure of 3,000 proteins, which means our scientists have to do the structure of 2,500, rest of the Japan 500, and it's a 5-year commitment. And they have to finish, and they finished ahead of time, actually. So it was running 24/7. So all we could do is do some prediction of the, computational prediction of the protein structure, and we were motivated to go forward. So now the question is, what are these guys doing in the cell? These lab-made genes made from the intergenic sequence of E. coli. So out of 10 samples we selected, we lost 4 of them, but the 6 of them we could carry forward, and we found that one of the gene expression is actually resulting in lethality, lethal phenotype. So it's the cell numbers are decreasing, they're declining. So we said that if this is true, then if we now switch off this gene, we should be able to restore the cell growth. So that's exactly what we found. When we switched off the gene after 2 hours, when the cell growth is going down, we switch off the gene by removing the inducer from the medium. It was arabinose. So when we remove the inducer, we culture them fresh medium without the inducer. You can see the cell growth is going up again, thereby showing a linear correlation between the expression of the gene and the cell phenotype. So the story is not simple, though I tried to hide a lot of challenges and complexities. One of the things was that, the PBAT topo that we took, we did not design the vector. Therefore, the start codon and the stop codon, they're not at the edge of the vector. They are upstream and downstream, which means when the protein gets expressed, vector is also contributing its own sequence to the final protein. So which means that the black portion that you're seeing is contributed by the vector, the red portion by the sequence that we choose. So a lot of vector contamination, which again indicates that the phenotype that you just saw, ECA1 or here, this is not purely from the sequence that we have taken. Vector has added its own sequence. So we have to do something. Though we got the phenotype, but then we have to do something. So now we changed this vector system, and we took a PET expression vector and changed the entire process of installing the sequence. We considered the start codon, ribosome, and promoter along with the PCR, primers that we have to synthesize, and so using this strategy, we are able to reduce ECA3, which was earlier not showing any effect. It was exactly like the wild type growth curve. So here we found that when we are changing the vector system, the vector contamination is going to 0. Earlier there was no cell growth inhibition. Now cells are growing 3 times slower, and some biochemical assays suggest that it could be a DNA binding protein. Probably did we make a transcription factor or something? We do not know at the moment. Overall, the functional study of the lab-made genes can be seen when there's overexpression. At the moment, we don't have a knockout library of a non-expressed genome, because our studies indicate that your non-expressed genome is also having some potential to express itself. We do not know what's the purpose. We're trying to find the purpose, and this is something that is needed in future. The ENCODE project, the Encyclopedia of Coding Elements project, indicated that there's a tiny part of the genome that is proteome. The bulk of the job is into regulation, and some of it is not coding. So all we are saying is how many sequences can be encoded into functional genes and proteins. Now, when you ask this question, it becomes a combinatorial problem, because now from E. coli itself, you can have millions of combinations from the intergenic sequence. So when you have so many combinations, then you have to find out which combination will I test experimentally in the lab, because the search space is huge. So this led to the database issues and modeling and a lot of computational stuff, and that's where my PhD students were involved full-time into computation for many years. So we made a database thinking that the sequence that we are considering can be potentially a protein, a functional protein. So what would be a location of this protein? What would be structure, function, interaction, and so on? So now from there, we started experiments. Now we are more targeted, subset of the sequences. So from Drosophila junk sequence, or the intergenic sequence, so to say junk, we found a peptide which has absolute resemblance in terms of more than 95% resemblance in terms of the structure with a known antimicrobial peptide, which is alpha defensin. Sequences are different, structures are nearly similar. So when we found this, we were like amazed, because the sequence that we are coming from is from the intergenic sequence, which is never expressed. So now we said, okay, if we express this, what will happen? And we found that this is actually killing E. coli and Staphylococcus aureus and Pseudomonas aeruginosa. In other words, our computational prediction is getting experimentally validated. So then we moved on. My students chose 3 targets, MR1, EBM175, and MS119. And they said these are very good malarial targets. And then Professor Shailaja Singh, who is a malarial expert, she was at ICGB, she did all these experiments, and then she found that in presence of these peptides, we are able to get some growth inhibition in terms of the parasites are not able to enter the cells. So there is a bit of wall created between parasites and the RBCs. And remember, these are all naturally derived peptides from intergenic sequences. Now, if we redesign the peptides, one can get a better effect. And that's what is the next plan. We also asked, what if tRNA? This was a totally crazy idea, because things are working slowly now, we're getting good results. And now we're emboldened to ask different questions. So we said if tRNA had the ability to translate its sequence, it was all the time doing an outreach program for millions of years, it's still doing it. Get the amino acids, put them to ribosome interface, and polypeptide is made, never care for itself. So we said, if tRNA sequence is itself translated, what kind of peptide would be made? Of course, we can't do this in the living system at the moment. What we did is we did a lot of bioinformatics initially, and then selected the right kind of sequence, and then we went on to do the experiments. And we tried a lot of cell lines, a lot of microbes, and parasites and things like that. This was also done in Dr. Shailaja Singh's lab, these kind of experiments. And so she found that in presence of these peptides, the TRAPs, at 30 nanomolar concentration, we are able to get a very strong anti-leishmania effect for this particular strain. And so this is currently in publication in the revision process. And so this was another source of therapeutic peptides, which has just suddenly come up without our conscious participation. It was just a random idea. We are also trying to make more and more molecules. We have got anti-Alzheimer peptides, again from intergenic sequences. And I was talking with many scientists from US, and we were discussing the possibility of making more proteins and peptides. So we are now very close to making peptides from different sequences which were never allocated the job of transcription. For example, antisense sequences. Introns is partially transcribed but never translated. So repetitive sequences and so on, and pseudogenes, so we are now trying to revive them and say that if something can happen. 2009, this publication came up. In 2010, I was invited by European Science Commission to give a talk. And I want to highlight this particular aspect because people are always after impact factors. The paper we published, first of all, I wanted to publish this paper in my own journal, which was Systems and Synthetic Biology. And I sent to Ron, he rejected the paper. He said, no, there's too little data, so you work a little more. At the time, I had lost my job and I had to come to India in search of a new job. So we had no funding, no group, no publications or the data that he was looking for. So we submitted this paper in another startup journal, which was barely 6 months old, called Journal of Biological Engineering. And, after some minor revisions, he accepted this publication. And it has zero impact factor. It was barely indexed in the PubMed. But we were delighted when we received the invitation from the European Science Commission for the talk. And then it kept on and on going forward with US National Academy of Science, IUPAC. You've seen the IUPAC formula that you read in chemistry. So they were also keen to know. Most surprising invitation came from Organization for Prohibition of Chemical Weapons. You can see this building, OPCW. It's like a parliament. And, I was pitched against 5 more speakers. And you have to address diplomats from 80 different countries. It's not a science event, it's more of a diplomat event. So, and I was representing Asia, and one of the questions that came up, in fact, I was surprised, why did they invite me? This was a bit of surprise there because not only the invitation itself, but also because somebody asked, can you make a lot of genes? I said, yes, it's possible. And then the real question came up. He said, can you make a bioweapon? Well, I said, look, you made me nervous, but you also given me ideas. So why don't you give me funding? So on a lighter note. But then we were very conscious that whatever we make should not be from the viral genome. We should try to get into the therapeutics area. Will be safe in terms of our ethics and also the applications. And then the other things went on. So this led to spinning off a company called Foresight Biotech at JNU with my postdoc, who was also from School of Life Sciences, Siddharth. So he's running this company, and we are hoping to deliver molecules for the pharma. We have a small inventory. The best part is that, you name the molecule after your name, because these molecules were not made by nature. At least the database that we are familiar with. So far, these are brand new. Everything started from, so to say, junk, and there are junk genes, junk RNA, junk proteins, and so on. So sometimes my students like to call this new area as junkomics, a new way of doing biology. So this is our small offering, as I was mentioning a while ago. We're also into policy aspect. There was a 1 year of project that we had taken from DBT, and we have submitted the report to DBT. Because there's no policy on synthetic biology in our country. It's a self-regulated system, but we did this foresight study leading to a policy framework, and there's a lot more to achieve in terms of regulation and responsible innovation. So we have joined the Global Biofoundry, Binay and I. He's also professor at School of Biotechnology in Genomic Sciences. So this would be, of course, our future interest. And these are the people who have initially been very generous to support this entire idea. And now Dr. Manju Jain is offering her support, and we are working towards delivering some products. Let's see how it goes. So these are the people. I really like to thank Professor Sakaki, because this is amazing. He gave me a couple of $1 million, and he said, "Look, this is the money given to you. Do what you want. You just do good science. Don't worry about the UCSC and the other procedural things." He gave me 2 secretaries. He said, "They will do all the work for you. You just sign the paper, do good science. Wherever you want to go in the world, wherever you want to send your students, just do that. All we want is good science." And the way it was managed there was, you have every year you have a contract. That means you don't have a permanent appointment. Your appointment is only for 1 year, and every year your appointment gets renewed based on your performance. So how they used to review was that at the end of the year, when there is Christmas time, if you're done well, the director invites you for a dinner in a nearby nice decent hotel. All the PIs who are working in that institute, he invites for a dinner, and then he says, "Okay, good job and good luck for the next year." And then you realize, okay, this means continuation of my job. And that's how we just enjoyed working here. And if I would have to write this project to DBT and show some initial results, it would have never been funded, trust me. Even now also I get trouble when I write to them. They have 1 million questions, and they are mostly worried about the UCSC more than the project goals. That's how it goes here. But the way it happens overseas is the productivity is everything. You got to walk the talk. And so there were challenges initially. People did not accept us the way we were trying to propose it. There are a lot of oppositions, a lot of ridicule and so on. But nevertheless, we survived, and we reached the present stage. We always receive motivation from the small atom that says, "Be positive," even if you're always surrounded by negative things. So thank you very much for your kind attention. I'd like to stop the presentation and go back to the discussion. Just a moment.
Yeah. Thanks a lot, Professor Dhar. It was a wonderful talk, and definitely you took us on a trip into newer possibilities at the interface of biology and the non-living. So that's really wonderful, at least to us. I mean, these are newer things for us, and I'm hoping students of biochemistry specifically, they must have enjoyed the talk. And I really want the students to come up and put up some questions. In fact, Professor Dhar, I would really request, because I see in your CV and as you spoke, how you started and where you are. I mean, it's a successful journey, and at your own terms and conditions, the things which fascinated you, without worrying about the publication component, the UCSOE component. So that's really amazing. So on that note, maybe it would be really great to have you sometime physically in a university, or maybe because with respect to, because you see a lot of sound around with respect to innovation and entrepreneurship. It's not a one-day job. It has to have a solid ground. So and our policies and we are pushing students into that direction. But I think it has to start somewhere. It has to nucleate somewhere. So that way, it would be really great if you actually come here and have more, interaction with the students, or we can pick up some students and come to your place. So something we are already in an MOS with the SBT JNU. So I think we would really like to know more about the area, and it's really, really fantastic and exciting, at least to me. So yeah, students, please, I would like students to come up with some questions.
Sir, Professor Dhar, yes, namaste, sir. So, sir, I have one question. So, there is some fine line between the genetic engineering and synthetic biology.
Okay. Well, you see,
what comes in the genetic engineering and what comes in the synthetic biology? So
Right.
if you can, because you are expert in this area, so I can also tell students sometimes, what is the synthetic and what is genetic.
Well, you see the intent. Right, sir. Right, sir. Thank you very much. That's a very important point because intent was to engineer genetics, but that was never achieved. It was all a probability. It's like you throw the bricks here and there and you hope that some of the bricks will stack on each other and make a wall. That's the way we have done, so to say, engineering in genetics. And we call it genetic engineering, which is actually a misnomer. We should call it recombinant DNA technology. That's okay. I can live with that. But to say engineering would involve standards, would involve rules of composition, would involve construction from the scratch. And the way engineers enjoyed the standards built by physicists early on in the 19th century, they did not have the standards. So the concept of standards has come in, but the trouble is that we cannot use the standard of the level of engineering. Because we're looking at analog and nonlinear systems, while as engineering systems are essentially linear and digital. So we have to have a different approach. Maybe it could be, random mutagenesis and directed evolution could work sometimes much better than rational design approach. So this would be my take, because synthetic biology essentially involves building the standards, and we don't have those standards, even though people are practicing that. Once we have the standards, and I'm talking about composition standards, that means, suppose you say that, for isolating a part, which is a gene, this is a method. Now, for isolating the gene, 50 different labs will have 50 different variations of the techniques. So which one is the standard? We have to first fix the process standards. And then the behavioral standards. Say, for example, if you isolate protein in your lab and it's an enzyme, and you do some measurements and you say, okay, I'm going to find out the turnover rate or whatever constant. And I do the same thing, because the purification methods are different. So you will get different values, I'll get different values. That's how we saw in systems biology, when we had to build a virtual cell. So standardization of process and standardization of component, once that comes in, then the true engineering will happen. I'm sorry, this is a long answer to your question.
No, it was fine actually. So probably because one of our courses, like the name is genetic engineering. So maybe in future we can change to recombinant DNA technology. Because second semester students are genetic engineering as elective.
Maybe students will get depressed. Maybe for students we retain genetic engineering, but in practice it is recombinant DNA.
Thank you, sir. Thank you very much.
Otherwise they will get depressed.
Thank you.
Yes, students, please. It would be great to have some questions from the student side. Yes, Evelyn, you please, you can switch on your camera if you're okay with that.
Yes, ma'am. Yes, ma'am. Sir, I have a question. Sir, using genetic engineering, is scientifically possible to reverse the aging?
Now you're asking me very difficult questions, which are beyond.
Sorry, sir. I read news about the reverse aging. Jeff Bezos gave you funding on this project.
Right.
So I'm curious about that. How is this possible?
See, this is not this is an important question, but at the same time, the pursuit to reverse the aging started way back early 90s. If you look at the publications that were coming on telomerase enzyme, there was a phase when the telomerase enzyme was discovered, very early 90s, if you see a Cell paper there, what people found is that, well, you know the story, I'm not going to repeat this entire story, but what I'm trying to say here is, in very brief, people thought that by synthesizing telomerase enzyme and adding that to the beauty creams, the ointments and the lotions and the tablets, they can increase the aging, or they can keep the skin healthy. Okay, they can reverse the aging and keep the skins healthy. And there are a lot of companies. If you look at mid-90s in the US, there are many companies spinned off on telomerase enzyme. They add telomerase to these ointments, and they were marketing as the beauty products. It did not succeed. because people found telomerase independent mechanisms of elongating telomere. So it is not just one component, it's a multifactorial thing. So aging is a multifactorial. It's a huge complex there. Whether we are able to get it right, I think my take on this would be that if we know how to accelerate DNA repair, if you make DNA repair more efficient, you may have less dead cells. Okay. So we need to really think of DNA repair. Our DNA repair is efficient, but it does not reach every nook and corner of genome. Like, for example, we are talking to each other today and the tube light is on. UV rays are falling on the skin and damaging the DNA while we are talking. And you have your DNA repair system taking off these, broken parts. But then it does not attend every nook and corner of genome. It leaves some of the genome unattended because it cannot be so efficient to scan the entire genome. So we need to work on DNA repair system. Yeah.
Okay, sir. Thank you so much, sir.
Yes, more questions?
Yes, sir. Yes, ma'am.
Yes,
गुड इवनिंग सर. सर आई हैव अ क्वेश्चन. सर जैसे कि कंस्ट्रक्टिव बायोलॉजी में या सिंथेटिक बायोलॉजी में आप जो बात कर रहे हैं कि हम बेसिकली उस पर्टिकुलर थिंग्स को पॉइंट आउट करके एक्टिव करना चाह रहे हैं जो अब तक हमें पता नहीं चला बेसिकली. तो ये तो नेचुरली अब तक नहीं था ऐसा, हम सिंथेटिकली ही करने का प्रयास कर रहे हैं. तो सर कर लेंगे फ्यूचर में, बट सर इसका नेचर पे क्या इम्पैक्ट होगा, नेचुरल इकोसिस्टम्स पे क्या इम्पैक्ट होगा, कुछ चीजें नहीं आएंगी तो नेचुरल इकोसिस्टम पे इसका किस तरीके का प्रभाव होगा, बेनिफिशियल होगा, हार्मफुल होगा. हम सिंथेटिक बायोलॉजी ला रहे हैं तो ये मैं जानना चाह रहा हूँ कि ला तो रहे हैं बट इसका नेचर पे क्या प्रभाव होगा. नेचुरली ये बंद थे, ये चीजें नहीं थी.
आपने बहुत अच्छा प्रश्न पूछा है. Once again, thank you very much for lovely, questions I'm getting today. So Rakesh, in response to your question, there are two aspects here. One is making the molecules. One has to be very clear about distinguishing it. One is making the molecules, and another is making the organisms. ठीक है ना? जो आप बात कर रहे हैं वो ऑर्गेनिज्म लेवल पे बात कर रहे हैं. अगर हम नए ऑर्गेनिजम्स बनाएंगे उसको बायोडाइवर्सिटी पे क्या असर होगा. अगर आप नए मॉलिक्यूल्स बनाएंगे, वो तो वैसे भी सिंथेटिक केमिस्ट्री में हजारों मॉलिक्यूल्स बनते जा रहे हैं एवरी डे इन द इंडस्ट्री. सो इट्स नथिंग न्यू. मेकिंग न्यू जीन्स फ्रॉम एन एग्जिस्टिंग सीक्वेंस, well, will it go against the nature or not? We are not putting it back into organisms. We are making products from here. So it's like we are saying nature has already made the sequence. We are trying to find the purpose of this. It's like annotation of a next level, functional genomics 2.0, wherein the not coding sequence or non-expressing sequence is made to express, and you find a useful function for industry, for health sector, for energy sector. You make a new enzyme. Like, for example, we are trying to make an enzyme, a restriction enzyme from intergenic sequence of E. coli. We have found some sequences and we have done some initial experiments. Now, it will not go against the nature. Making therapeutic peptides will not go against the nature. It will only add on to your toolkit of handling the diseases. But your question assumes a very, very big importance the moment you talk of redesigning organisms and putting them back into nature. So that is where there's a big issue of biodiversity, because people are now saying that we can, we can bring back dinosaurs if we have the genome, and there are genomic information available. They are saying we can do a lot of those organisms that have been extinct. We can bring them back. I would say it's not impossible. The technology of long DNA synthesis has gone so fast that you can order a genome. Let's say a microbial genome. You want to order a microbial genome, you need a couple of lakh rupees. But of course, there will be some biosafety and security checks. But I'm saying the technology has gone, that much advanced that you can bypass the recombinant DNA technology process, and you can. And nowadays, people are doing it. We are also doing in our own lab. That you design the vector on a computer. You don't do a recombinant DNA vector construction in the lab. You design in the computer. And then whatever edits you do, put markers, put genes, put everything. And then save it as a text file, ATGC, and all that. and then send this to a company. It will chemically synthesize and deliver you a recombinant vector in 3 to 4 weeks. What would take years to gather? 3 to 4 weeks you have this innovation. Well, it is acceleration of innovation, but there's a lot of discussion going on. It is led by the Center for Biodiversity in Canada, and it's a global organization. It's a mandate of United Nations to have all the countries on board to discuss the possibility of the misuse of synthetic biology or the responsible innovation. That's where you need the policy, and that's exactly where we have made a contribution and sent the first draft of the policy framework to the Department of Biotechnology. So I would, briefly therefore, our work does not go against nature because we are only making the products from nature. We are not creating a brand new DNA. For example, people are creating non-ATGC DNA. Are you familiar with this work? There are people in Japan who are saying, well, these the current DNA is more than 3 billion years old. अरे यार बहुत पुराना molecule है. अब कोई नया DNA बनाओ. तो उन्होंने non-ATGC DNA बना दिया. अब उसको functional बना रहे हैं. वहाँ पे हमें थोड़ा सोचना पड़ेगा. People have made, instead of 4-based DNA, you have 6-based DNA. And this is functional. It's replicating. So does it go against the nature? Yes, to some extent, yes. As long as it doesn't go in the wild, if it remains in the lab for only research purposes, it's fine. But the moment it goes to the wild, then there'll be 10,000 regulations and it will not be successful there in the current form. So one has to be having a policy on that.
Thank you, sir.
Okay. Thank you very much.
Thank you, sir. So I think the way discussions are taking the directions, a lot of food for thought is being, given to almost, and I'm sure many students will be having many questions, but most of them will shy away and will not ask.
So Dr. Manju, I have one more question. If
yeah, Dr. Ravindesh. So, sir, Dr. Ravindesh is our colleague, faculty in Department of Biochemistry itself. Yes,
yeah.
Sir, please.
Professor Dhar, thank you, sir, for such an informative talk. I really enjoyed it. My question is not biological. My question is more on the fact that, the proposal that you gave, did it start out like a side project? Because, convincing people of something like that, you are going to make non-coding genes code for a protein or for a tRNA to act like a peptide is not going to be easy. And that is something very difficult, to convince, like you said, that even if you write a DBT project, probably they won't give you a grant on that. So my question is, what gave you the confidence to go ahead with this? Did it start as a side project? And what would have happened if you had failed in, proving your point?
Wonderful. I think that's a turning point of our career. So what was the trigger? The trigger was faith by the president of organization, who was also president of Human Genome Organization, Professor Yoshiyuki Sakaki. It was a pure faith he had in me. So he said, use this funding and do something useful. What you want to do, how you want to do, you're the boss. We'll not question you. That leap of faith was very, very important. It was very critical. We could have failed. Actually, if you look at the experiment, if we would not have got the phenotype, the cell death phenotype in the first experiment, out of 10 combinations, we lost 4, and out of 6, only 1 gave us phenotype. If that wouldn't have come, probably we had to close the project because, we were running short of time. It was already 1.5 years and we have to show something. And running from embassy to, other places in search of the funding, it would have been very difficult. But we were very fortunate to get this. Now, coming back to India. In India, the way the funding happens, somehow people are scared of innovation if it is too radical. You need to have a clerical mindset that you will do incremental innovation. You will not do a radical innovation. Well, for radical innovation, you have DST funding. But somewhere, I'm not so happy with the DBT model, wherein you need to be in the safe zone all the time. So if I have to submit the project with DBT, it would have been easily rejected. In fact, I can also share with you that when I came, I had come with a collaboration from MIT on a platform called Genome Designer. And we had not only submit the project, but we also gave them the screenshot of what we have made, because we had spent 6 months with TCS in building the first version of the genome designer, and they rejected the project. ऐसा तो कभी हुआ ही नहीं है तो हम कैसे करें? And at that time, I was in private university, and they didn't like the fact that, private university people can also think. So they rejected the project. And 3 years later, when I was the chair in some DBT committee, people came to me, "Sir, can you submit that project? It was lovely." मैंने कहा वो चला गया. MIT people have lost interest. So about this project, they would not have funded it. And we received motivation from this leap of faith and the results that we got in Japan. And for the last more than, I think, 11 or 12 years, we have been working only on one project, just one idea, nothing more than that. Policy to abhi hua tha 1 saal ka, wo to alag hai, but we did not change the track because there is so much to do. Imagine, 1.5% of protein coding genome gives rise to thousands and thousands of PhD theses. E. coli itself, it's such a small bunch of genes, giving rise to PhD theses in terms of hundreds and thousands. We are talking of converting 98.5% of human genome into proteome. Now you can imagine the scale. So there's a huge scope. It is just that one has to accept if you're not successful sometimes. But we just keep doing it.
Thank you. Thank
It's
you so
difficult,
much.
but then we are so excited to.
No, that's true, Professor Dhar. I really enjoyed listening to you, and I would actually like to share that I also listened to one of the talks by Shinya Yamanaka, who got the Nobel Prize for IPSCs, and he also had the same thing to say, that when he was initiating that project, people used to call him that you're going to finish your career by doing this, and nobody's going to you won't be able to achieve success in that. So you also had that leap of faith. And I also now, I'm remembering Barbara McClintock's story, because when she came up with this jumping genes idea, people again ridiculed her. And later on, so one has to, take leap of faith to make radical discoveries in science. So, thank you so much, sir.
Thank you very much. In fact, I was talking with Martin Shelfie, who received the Nobel Prize in GFP. We had hosted his visit when I was in Kerala. For 1 week, he was with us. So I gave this presentation there. And then he gave me a new idea. Instead of beating this idea to death, that is what our people do all the time. He said, he suddenly kind of a spark came up, and he said, would you consider making genes from the antisense portion of the genome, which has not been explored so far, using the same cassette? We said, wonderful. So we have started working in that direction. We have also, I did not present those results because of lack of time. We also asked, can we reverse the protein coding sequence? In the reverse of protein coding sequence, in the normal frame, maybe it's in a certain frame. If you reverse that, can you make a new protein? What we call as reverse proteins. We are working towards validating those results, and a lot of failures. A lot of people left the lab, and funding issues are there. But we are able to get funding from somewhere. So the fact that we started a company, gave us another source of funding, and that's the message I gave to my students. JNU being a place where it was next to impossible to start a company. And people used to say, yahan pe left ka bahut zyada hai, communism and all that. That's fine, no problem. So we made the framework. My colleagues were involved in that. And then we incubated the first biotech company. And it's been 3 years, and nothing wrong happened. And in fact, a lot of people got trained, and they also understand that the future model is not getting PhD postdoc in the US, coming back unemployed. That is not the way to go forward. In fact, we tell our own PhD students, abhi tumhare paas time hai, let's go to the nearby shopping mall and work there for part-time 1 hour a day. Don't depend on government to give you a job. Don't depend on anybody to give job? Can you get your own job? Can you make your own money? That's the way they do in foreign countries. They work in restaurants. We have to get into that culture because we are like very traditional and very safe zone kind of people. And if we don't get anything, then we start complaining. So you will not believe our master students, couple of them who understood what we are talking about, they go to the nearby shopping complex and they spend their weekends making some pocket money. And this gives you freedom and independence. It also takes the shyness away because once you're out of PhD, then it's going to be a harsh world. So we better get prepared. And so this is the best time to prepare. Yeah. I'm sorry. I think I'm running out of time. It's already taken a lot of time.
It was wonderful listening to you. And, you have actually given us another idea that we should invite you again for talking about the company that you have set up. Because entrepreneurship is another thing that...
Dr. Ravindesh, we really need guidance on those perspectives, which we are talking a lot about, right?
Yes.
So, will surely be. And with this, I would request Dr. Ravindesh to maybe he can give a formal vote of thanks. And sir, definitely we are looking forward for a physical meet here. And maybe, involving students rigorously, because I see you have lots and lots to give these students, and we are young, people trying to, understand the rules of the game still. So you will be a great mentor on that aspect.
Thank you very much. I'll be delighted to contribute. We're also at the learning phase. It's not that we have made something big, but I must say that we have spinned off two companies. One is this JNU company, and the other one is another company outside of JNU. This is on cultured meat, because there's a big global industry on going away from slaughtering animals and creating a cultured meat. And also there are many aspects to that. So we are probably in future, we might create another company and another one. So this is where we want to go in future. I'm talking to one of my colleagues who is an expert in AI, Professor Binay Panda, about him, his slide I showed in Biofoundry. So he's an AI expert. And so we're trying to bring AI into synthetic biology and do something there. So you might spin off a couple of more companies in future. But that's where you need more students, At the student level, it's easy to take a risk because you're still, 3-4 years, you get your scholarship. After that, it's a little tough. But that's the way it is. So, yeah.
Yeah, all right. Thank you so much, Professor Dhar. I mean, again, it is very interesting that you're talking. You've already started a couple of companies and you're thinking of starting another one. I think that's all the motivation that all the students and the audience, including faculty like us, need for our future. So I'm sure that everyone enjoyed the informative talk by Professor Dhar as much as I did. I think there were some very interesting things that he talked about. He talked about that Singapore technical assistant, lab assistant, who agreed to work on this project, and the PhDs and postdocs who did want to work on this project, right? So I think this gives us an idea that rather than going down a beaten down path, if you take a road less traveled by, that is something that will really make a big difference. And the tRNA encoding a peptide, that is like turning the biology on its head. So who would have thought of that, right? Making the non-coding components code for a protein. So lots of interesting things in his talk that I personally enjoyed. So, now it is my privilege to offer the vote of thanks for this event. And first and foremost, Professor Dhar, we are really grateful to you for taking time out of your busy schedule and sparing almost 1.5 hours with all of us here at Central University of Punjab. And since many of the attendees are actually aspiring researchers, many of them are master's students, so I'm sure they would be inspired to take up synthetic biology maybe as a research topic in future. I would also like to thank our Honorable Vice Chancellor, Professor R.P. Tiwari ji, for encouraging the faculty of our university to invite esteemed researchers for academic talks like this. So such initiatives expose the students to what is really happening, what is latest that is happening in scientific research, and they get to know about science beyond what they probably would read in their usual textbooks. I would also like to thank our DIA, Professor Ramakrishna Vusurika, Professor Anjana Munshi, our Dean Research, for taking the time out and joining in. Also the faculty members from other departments, from our department who joined in. And last but not the least, thank you, Dr. Manju Jain, for organizing today's event. And also thank you everyone for joining in. So thank you.
Thank you. Thank you very much for having me here today.
Thank you, sir. Looking forward for a meeting soon and to have us in Central University of Punjab. Thank you, sir.
Thank you.
Okay, sir. Okay, thank you, sir. Thank you.
Thank you.